right, now, one last category of regulatory RNAs. Once again, there are many others, most of them undiscovered yet. More on that later. But this is the long non-coding RNAs. Or LNC RNAs for short. Okay, what are they? Well, long means they're at least 200 nucleotides long. Most of them are many hundreds, even thousands of nucleotides long. Non-coding means they do not have the instructions for for making a protein like messenger RNAs do, and things like that. Okay, now you find these guys scattered throughout the genome. In vertebrates, it's estimated that there are several hundred, perhaps even 1,000 of these guys in our genome, different LNC RNAs. And they've been relatively recently discovered, but they seem to have a lot of potential interest. What do they seem to do? Well, people have done experiments where they deleted the gene for a specific LNC RNA. And what happened is you knock out one of these genes, delete one of these genes, and what happens is a whole bunch of different other genes are either overactive, overexpressed, or underexpressed. So the same RNA is going to ramp down the expression of some genes and increase the expression of others. So they seem to play a role in regulating the expression of multiple genes. Now, how do these guys do that? Well, that's where we're a little bit more theoretical ground. It's a little bit more shaky as the mechanism. The widely believed mechanism of this is that these guys act sort of as scaffolds, as assembly points for proteins involved in regulating transcription and other things. In other words, instead of the proteins just sticking to each other, they're sort of assembled onto this LNC framework, and that holds them, the right proteins in the right place to regulate the process of transcription. In other words, the use of a gene to synthesize a messenger RNA. So, so they seem to act perhaps as scaffolds that actually help to assemble the appropriate proteins together to do a process like transcription and stuff. Now, the process will occur without these guys, but may occur either too much or too little depending on the particular gene you're looking at. And here again, if you've got several hundred of these in the genome, and each one of them can handle and regulate transcription of a 10 or 20 or 30 genes, that suggests that a good portion of our genes are regulated in part by LNC RNAs as well. So it's clear RNAs are playing incredibly important roles in regulating the genome and its use. Okay, now, another interesting thing. You can use what we call antibodies. These are proteins produced by the immune systems of animals. Label them with radioactive elements, fluorescent dyes, or whatever. You can use that to localize certain processes in cells. So you can make an antibody against a protein of interest and say, where in the cell, no pun intended, where in the cell is that located? Now, let's suppose we do something and we have some of the key proteins like RNA polymerase or something like that that's involved in the process of transcription. So you look at the cell and you ask, where are these things? You look in the nucleus and you find that these things are located in discrete spots in the nucleus. They're not scattered all over the place. They're only found in discrete spots. Almost as if there are sites where transcription takes place, and you move the appropriate gene to this transcription factory, and there it happens. Okay, now, so this is where, say, RNA polymerase is. This is where transcription is taking place. Now, let's get some labels for L and C RNAs and see where they're located at. And lo and behold, we find a whole bunch of them localizing in the same place as these transcription factories. That's also telling us these guys are important in regulating transcription. They're found in the same place that transcription takes place. Now, another cool thing. Let's suppose instead of looking for the problem where transcription occurs, most of us who took genetics probably know that in eukaryotes, messenger RNAs undergo a lot of processing before they become mature messenger RNAs. So you get this mRNA precursor that's transcribed off the gene. You have to do things like cutting out what are called intron sequences that don't have instructions for making the protein, and then splicing together these exon sequences that do. It's kind of like cutting the commercials off a tape of a television show. Okay, now that RNA processing is critically important. And there's a whole bunch of different enzymes and stuff that do that. Now, once again, you can use antibodies to localize where these RNA splicing centers are. And here you see another speckled distribution of where splicing occurs. So we have transcription factories, we have splicing factories in discrete areas of the nucleus. Once again, using certain L and C RNAs, guess what? We find they co-localize. In other words, they're found in the same place. So it's suggesting that L and C RNAs help assemble the splicing machinery together and hold it in place. So it's telling you these guys are important. These guys are very important. They're going to regulate transcription, each one of them, of multiple genes, and they probably regulate splicing as well. That's pretty darn important. Okay, these are a couple of the major categories of regulatory RNAs, but there's more. We're not going to go to any more categories. There are a few other ones that play lesser roles that are currently known about and stuff. But here's an interesting thing. Here's an interesting thing. Let's look at the human genome, a good example, right? And this is typical of vertebrates in general. Couple things about it. We've got 3.2 billion pairs of bases. That's a lot of DNA. Just writing that sequence out in the standard one-letter code for each base would fill up 600 copies of your textbook. Or you'd probably have to put it on a DVD. I don't think a CD could hold that much information. DVD could. Okay, that's a lot of information. In that, we have about 22,000 genes. Okay. Now, if you take the total DNA on all of those 22,000 protein-coding genes, plus the control sequences, plus the introns, plus all that stuff, take all that stuff together and add it all up, and you find it's only a couple percent of the total DNA content. In other words, only about 2 or 3% of our genome is actually stuff related to protein-coding genes. That means the gene itself, the intron sequences, all the control sequences in front and sometimes behind the gene and so on. So it's saying that 95+% of our genome has nothing to do with protein-coding genes. Well, what's all that DNA there for? It's a huge amount of DNA. Why do we have it? Well, in numerous organisms, especially in vertebrates, we're finding something that is amazing. And that thing that's amazing, has been looked at in multiple organisms, is at least half, and often the majority of that genome is transcribed. In other words, things are read, and you make various kinds of RNAs off of that. Yet only a few % of the genome are actually coding for messenger RNAs and their precursors. And you throw in the other known categories of RNAs, that's a fraction of a % more. So, since making RNAs, the process of transcription, uses a lot of energy. After all, you're doing covalent attachment of one nucleotide to the next. That's going to use a lot of energy to make that. That's condensation reactions. Why is all that stuff being transcribed? Why are all those RNAs floating around in the nucleus? Well, that's the future of RNA biology. It must mean that a huge portion of those RNAs are undiscovered yet. They must be there for a reason. Why would you waste expensive nucleotides, energy and stuff, making various kinds of RNAs that are an absolute waste? Even if you did it by accident, evolutionary pressure would be to stop making stuff that has no use for you, because otherwise you're just wasting resources. You're wasting more resources than the federal government does. Okay, that's pretty bad, right? Okay, so all those different transcribed RNAs must be doing something, and something important in a cell. Otherwise, we wouldn't have any reason to make them. What are they? We don't know. It's telling us there must be a whole bunch of undiscovered categories of RNAs that are playing functionally important roles in cells, development, whatever, that we know nothing about yet. Which means the RNA biologists are going to have a lot of work for a long time to come. Figuring out what these RNAs are and what they do is a wide open field. Another interesting thing about this, looking at the genomes of numerous different organisms, everything ranging from single-cell protists all the way up to human beings, you can use computer programs and say what parts of the genome are very similar or identical from one organism to next. And when you have things that are extremely similar from one organism to next, we call these things conserved sequences. Now, one case in point, we mentioned about that before. Those two little pieces of that large 23 or 28S ribosomal RNA, they're absolutely invariant, the same in every organism from bacteria to our cells. The rest of the molecule varies quite a bit, but those sequences are universally conserved because that's the active site of the enzymatic activity that joins amino acids together, changes single nucleotide, and that whole process grinds to a halt. So you look in the genome and say, what's the most conserved sequence in the genome? And they identified hundreds and hundreds of things that are very similar or absolutely unchanged from one organism to next, including organisms that are widely divergent, like protists in humans or arthropods in humans. Some of those sequences are associated with the genes for highly conserved proteins. There are some proteins like the histone proteins that organize DNA and help to regulate transcription. Those are very similar from one organism to the next, almost identical. But a good portion of the most conserved sequences around are not anywhere near protein coding genes. Some of them have been identified as associated with L and C RNA genes and things like that, but others are there, but we have no idea what they're there for. Yet, if it's something that's virtually unchanging from one organism to the next, it's got to be important. It doesn't mean those regions don't undergo mutations. It means if you undergo mutations in those regions, you're toast, you're dead. It's absolutely something you cannot mess with. What are those? Nobody knows yet. But it tells you the field of RNA biology is just beginning to open up. There's a lot of stuff out there with RNAs that we have no idea what they do. But they must be there for a reason. It must be very important. So who knows? come back, your kids go, I hope I'm retired by then, but if your kids come back and are still teaching cell biology, we might have a lot more categories of regulatory RNAs that have not been discovered yet. Okay, well, that covers RNAs and a little bit about RNA biology. So we can clearly see RNAs play critical roles in genetics and the regulation of genes.